So thanks everybody for being on our July call, and sorry about the delay last week. I just got so many emails from people they wouldn't be here because of the 4th of July holidays in the US, and I guess there's some other holidays going on, so I thought I'd push it back a week. So hopefully it's not too much of an inconvenience. So what I'm going to do is we got a couple things on the agenda that I want to cover today. One is I do want to hear Elizabeth's talk on the Gowanus Canal. There's a lot of data to cover there. And then I got Concetta from Oxford Nanopore Technologies to give us an update of where Nanopore is and all the new developments there. And then hopefully I can get either JJ or Krista or Horia to give us an update on just the small piece of work that they've been doing on the samples. And I know it's really just a matter of transferring samples, and we tested and retested this way to send samples overseas on a column. So maybe we can just get a little bit of an update on that. So without further ado, I think what I'm going to do is hand over the screen straight off to Elizabeth and let her take it away and give herself an introduction, and I will make you the co-host. So you should have the screen.
Cool. All right, share screen.
And please introduce yourself, because I'm not sure everybody knows who you are.
Yeah, sure. All right, let's see if I do this. Does it go full screen? Uh-oh, no. Wrong share. Present. Cool. Do you guys see gray text, black backgrounds?
Yep.
Awesome. I'm gonna make all of y'all's faces kind of bigger so I can see you too. There we go. So my name is Elizabeth Hénaff. I'm an assistant professor at the NYU Tandon School of Engineering. I did my postdoc in the Mason lab, so I see a lot of familiar faces and familiar names on this call. So I was a postdoc at the Mason lab until 2018, and then I joined NYU here at the engineering school. And I'm in a kind of particular position because I'm a computational biologist by training. My appointment is in the design department at the engineering school. And so I work on projects that are kind of at the intersection of both biology, design, and engineering. And so here at NYU, I founded the Laboratory for Living Interfaces. And so we have a whole portfolio of projects that is aiming to understand the relationship between the built environment and the built environment microbiome, with the understanding that if human health is really tightly related to our relationship with our microbiome, and the human microbiome is influenced by the environmental microbiome. And for most of us who live in cities, the environmental microbiome is very much sculpted by the design decisions of architects, city planners, material scientists, all of the people who have made the design decisions that have shaped the spaces that we inhabit. And so in general, I'm interested in using the city as a laboratory and finding interesting case studies, interesting urban environments where knowing something about the urban environment and then measuring the microbiome in that environment can tell us something about the relationship between kind of the abiotic material characteristics of that urban environment and the design decisions that led to it, and the way that that has influenced the microbiome. So one of these projects, one of these particular environments of interest is the Gowanus Canal. So the Gowanus Canal is a waterway in Brooklyn that has been that was used to be a creek that was dredged to become a canal, and that has been contaminated by industrial waste for the last 150 years. So that's just a little intro of what I'm going to be talking about today. And I like to use the term holobiont urbanism. The holobiont being a term that kind of encompasses the notion of host and symbiont, with the idea that you can't really dissociate the individual from the individual's microbiome. So, us, thinking about the notion of an individual as like a human body or a city is indissociable from the microbiome with which it lives in symbiosis. And so that's kind of the term that I've coined to describe the work that we've been doing. So as I mentioned, one of this particular case studies that I wanted to talk about today is the Gowanus Canal. So it was dredged in 1853, and since then has served as a de facto dumping site for industrial contaminants. So the factories in operation around the site have included coal tar factories, paint making factories, tanneries, cement plants. And this particular canal has accumulated all of the toxins that are related to those industrial practices. And in the early 2010s, it was declared as a Superfund site by the US Environmental Protection Agency. So meaning that the level of toxicity that it presents and the facts also that it's embedded in an urban environment makes it a priority for remediation. And so as such, it's an extreme environment in the sense that it contains really high concentrations of particular toxic compounds. So using the, very scientific sampling mechanism of 15-foot long PVC tubes and canoes and human-powered canoes, we paddled out into the canal to collect sediment samples. And so using kind of the same principle as, a bartender who samples a fancy cocktail by putting a straw in the drink and then capping it with the top of your finger and then pulling it out to sample the cocktail. Well, in a similar way, we used these 15-foot long PVC pipes, dug it into the sediment, capped it with a cap and pulled it out and collected surface sediment samples in that way. So I'm going to talk a little bit about what we found in these surface samples. And then also another set of samples that were collected by a collaborator who might be on this call or might not, Sergios Kolokotronis. And so he partnered up with some folks at the DEP, the Department of Environmental Protection, who are taking core samples, so down 20 feet, up to 20 feet deep, down in through the sediment of the canal. And so I'll talk a little bit about what we found in those samples as well. And so given the short time frame of this talk, I'm just going to do a little bit of an overview of the results that we found, and I'd be really glad to go in deeper conversation with anybody who's interested. But in particular, I think what I'd like to discuss with the group is kind of this idea, maybe more of an existential or philosophical question of what is it exactly that we can learn from these extreme environments and from the microbes that can inhabit it that'll help us understand how we can survive on our rapidly changing planet. So this is a map of the canal, and we have two different types of data sets. One of them is surface sediment samples, so collected from the canoes as I described, and one of them is core samples. So those surface sediment samples were collected at 14 different locations along the canal over 3 different time points. So summer, winter, and spring again. And then the core samples were collected at 2 different locations at different depths. So one core sample went down to about 18 feet deep, and then one core sample went down to about 11 feet deep. So when I say surface samples, I mean that the like longitudinal study of surface samples, and then core samples are the ones that went deep. So as a kind of summary of the results that we found looking at species classification in the core samples, we realized that the core samples grouped in kind of 3 different categories according to their depth. And so this is Kendall's correlation looking at species classification. And so what shook out there was kind of 3 different depth or 2 different depth measures, shallow and deep. And independent of the location of the core, all of the shallow samples looked the same, and all of the deep samples looked the same. So we do know that these cores responds not only to the kind of the depth characteristics itself, but also a gradient of chemicals. If we look at the time series data, also here looking at species classification, we can see that there's a slight shift in species composition over seasons throughout the canal. So here, if we look at this clustering analysis, we can see that there's a shift kind of between spring, summer, and winter slowly in the species classification. But generally the species, and also we'll see later the metabolic functions, stay pretty much consistent over the period of time that we sampled here in the canal. So maybe here of interest for this particular group, we found a bunch of different extremophile types of microorganisms in the canal. So, you know, this super fun site embedded in an urban environment can be considered a storehouse of extremophiles. So for example, psychrophilic or cryophiles, also thermophilic, which was kind of confusing. And I'm open to suggestions of interpretation with respect to this, because the canal is very much a marine environment, kind of a tidal estuary that receives input of brackish water with high tides twice a day. And then also halophilic organisms, which does make sense with respect to the fact that it's a marine environment. We also found some interesting alternative energy metabolisms, looking at the metabolic potential of these organisms. We found methanotrophs, chemotrophs, and also different types of phototrophic organisms. I'm super glad to discuss the details of all of this with folks, with whoever is interested to dig deeper into this. But what's particularly interesting to us and relevant to the context of this particular environment is looking at bioremediation functions. So what we're looking at here is a metabolic pathway analysis using the sequencing data obtained from these samples. So instead of using that data to match databases of individual species or species markers, we looked at to match it up with coding sequences of genes, and then assembled those genes into pathways using the Metapsych database. And so, so we looked so given the history of the canal, and in particular the main source of contamination being hydrocarbons from the coal tar industry, we took a deeper dive in specifically looking at the degradation of these particular types of compounds. So for example, we find cresol degradation, degradation of a lot of phenolic compounds, toluene, and kind of the subclassification of these different types of contaminants. And then what we did was try to see if we could relate the species that we had identified with the bioremediation functions that we had identified. And so this can be kind of tricky using short read data, because oftentimes within the 150 base pairs of a particular read, you'll either get a match to an identifying species or to a particular gene sequence, but rarely do you get a match to a gene sequence that you can also attribute to a species. So most of the functions, metabolic pathways that we found, we were not able to attribute to particular species. So for Concetta, who's from Nanopore here, I think that that's a particular use of long read data that would be really meaningful for us at this time. But if we look at this plot here, what we can see on the left side are species, and then on the right side are pathways. And here we can see we kind of connected the dots as far as which species encoded which pathways. So we can see, for example, some pathways are encoded by multiple species here, or some species are responsible for multiple different pathways. And so what's interesting here, and I don't have this figure here, is that we actually see this relationship between species and pathways changing over time and also changing according to the depth of the samples. And so what this kind of is showing us here is that there's a certain set of functions that need to happen, but who is playing that function can change both over time and also within the environmental context that we're sampling. So here then we also looked at kind of bioremediation pathways in general, and found some interesting detoxification pathways, such as formaldehyde assimilation or arsenic detoxification. And so in this sense, it seems really interesting because this environment of the Gowanus Canal that has been considered as like a forsaken environment from the human perspective, it's like gross, it's contaminated, it's nasty, we need to get rid of it in any way possible, just pour concrete over it and like call it quits. It turns out is a really interesting and also a really rich source for unique biological functions. And what I think is also interesting to understand here is that, this kind of microbial population would be impossible to engineer in the lab. And these are really interesting kind of biotechnology applications of these functions for toxin degradation. And we can engineer a microbe to do a thing or to do a particular number of things, but having a whole population of different species that can cohabitate together and that taken together can degrade this whole cocktail of different toxic compounds while also, not being susceptible to the ones that particular individual species can't degrade, is a really complex ecosystem and is something that would be really difficult to engineer from the top down.
Elizabeth, I have a question if you don't mind me asking. So is the Gowanus Canal still being dumped into? This is legacy, it's not an active cesspool of extremophiles then?
No, the only input at the moment is combined sewage overflow. So there's input of fecal microbiome, but otherwise it's a really stable environment. So it really hasn't been modified for the last 50 years or so. And the only industry that's still present at the Gowanus Canal is a cement plant. So it is pretty stable. And so also as far as kind of interesting mining potential, we looked at antimicrobial resistance. And so I would love the input from experts in antimicrobial resistance to help us interpret these graphs here. And so, but we did, so with the idea that, given that this is an extreme environment and also a very competitive environment, we wanted to see whether there was any particular interesting mining potential with respect to antibiotic resistance in this environment. And so I know that I'm pretty much out of time, but we did also look at biological gene clusters. So what we did for that was assemble our short reads into long reads, pull together all of our samples, assemble short reads into long reads, and then look at biosynthetic gene clusters to see if we could find clusters of genes that would correspond to novel secondary metabolites. And found a couple of seemingly novel, potentially interesting ones. So I'll also have to call out here that Chandrima Bhattacharya was working as a research assistant in my lab for the last year and is the one who did most of all of these analyses, to give her full credit for all of these figures. And this is kind of an abridged version of the presentation that she gave at Meissen Lab meeting last week. But all this to say is that, we feel that the Gowanus Canal is not a unique environment by any stretch of the imagination. So this map here is a map of the Superfund sites in the United States. This is definitely not specific to the United States. They're distributed pretty much throughout the industrialized world. And so as such, I think it's interesting to understand whether these environments that have been anthropogenic environments that we have, as human beings, have created, might hold some valuable potential to help us learn how, you know, how microbes have learned to adapt to these contaminated environments, and maybe help us learn how also us humans in relation to these microbes can help us adapt to those environments. And so as such, I don't know if y'all are familiar with the theorist Donna Haraway, but she talks really eloquently about living on a damaged planet, and she published a book recently called Staying with the Trouble, which I think is an interesting context in which to think about these relationships and responsibilities that we have with you know, non-human organisms. And so you can add that to your summer reading list. So with that, I will stop sharing. And I don't know, Scott, if there's time for questions.
Oh, sure. There's definitely time for discussions, not just questions. It's technical
Awesome.
discussions. And I just wanted to ask you about culturing, because I know you and I had started doing some culture stuff down there. And I didn't know if you followed through with any fungal or just bacterial cultures. That was my first question. The second question is are you finding, or have you found, even though they're not extremophiles, have you found pathogens in the Gowanus Canal?
Yeah, so pathogens as in like the regular ones that you would expect in wastewater, yes. Nothing weirder than that. And with respect to culturing experiments, this has been, you know, it was a BSL issue when I was at the Mason Lab, and it still remains a BSL issue here at NYU. So I haven't been able to set up you know, sufficient BSL safety lab in which I can culture unknown bacteria and fungi. But that's on the docket as far as future projects, in particular in trying to understand this relationship of who's doing what. Like we can try to reconstruct that computationally, but ultimately doing culture experiments is how we could get that answer.
Hi, I have a question about your pathway prediction. Well, basically, I'm curious how you did it.
How we did it?
Yes.
Yeah. So we used the tool Human2 from the Huttenhauer lab. So actually, we used Human3 from the Huttenhauer lab. Chandrima will correct me. And so that uses the MetaPsych database for pathway prediction. So we did that for general pathway classification. And then Chandrima curated a specific bioremediation pathway database that then she used specifically for the bioremediation pathway analysis in particular.
So how much refinement was necessary?
I'll let Chandrima answer that question because I see that she's on the call now.
Can you repeat the last part? I didn't get you.
My question was how much refinement after the initial pathway prediction was necessary?
For the bioremediation pathway specifically, is that what you're referring to?
In general, but if you have an answer for bioremediation, that would be also of my interest.
So for bioremediation, there is a database which I used to cross-refer and then which was helpful. But as for refinements, like if you're using the Human3 or Human2, I think the general standard is that you clean the data previously, and after that use a normalized CPM method. So a lot of refinement was not needed, but finding the pathway of interest is kind of difficult if you do not have a lot of understanding of the pathways you're looking for.
Okay, thank you.
I also have a question. Also, when I came late, I didn't know a little bit of the project. Hey, always good to see you. I think I'm very fascinated by these new gene clusters. And since now, since SARS-CoV-2, so many sewages have been sequenced, so much is happening right now. It's not for you like Christmas right now, there's just so much data. And did you saw one of your gene cluster already in other parts and can start making associations?
Interesting. So you're thinking specifically of the relationship between the between the CSOs
Yes.
and the canal.
Yeah, because now you have, like, with all these new sequences you just rushed in the last year, you maybe have, you're lucky and find something which you can make starting associations, because right now you have only one Superfund sites and one gene cluster, but maybe you find similar combinations somewhere else.
Yeah, you're right. But I guess the thing that's difficult for me to think about disentangling is, you know, if we're talking about the sewage input, that actually, to me, has kind of nothing to do with the fact that it's the Gowanus Canal. Like if you sample sewage, you know, in the Gowanus Canal or from a sewage pipe itself or from a sewage treatment plant, like really what's I guess specific about the sewage is the fact that it's sewage. And if you find it in the Gowanus Canal, I feel like those are actually pretty transient inputs that don't actually affect the Gowanus Canal microbiome per se in the long term.
I was thinking in regards of the substrates combination and gene cluster combination that you maybe can, you know.
Yeah. So what would be really awesome is just doing like a nationwide Superfund microbiome study. Like that's something that I think would be super exciting, because there we can start seeing, you know, examples of convergent evolution, for example. So, you know, similar adaptations to similar contaminants, but coming from different sources or different contexts. So that's something that I would be super stoked to look at. And if anybody wants to like look at their favorite local Superfund site, I would be really into collaborating on that.
Okay, I recall the samples.
Yeah.
Very bad.
Yeah. Extreme Superfund, or like Superfund committee within the Extreme Microbiome Project.
More questions, and technical discussion too. It's not just questions, but if you guys want to talk about things, this now is a good time.
I mean, you know, actually, I have a question to put forth to the group. So one of the struggles that we've been having with these samples is getting EHS to sign off on us being able to handle them, period. And so I guess maybe this is an interesting question for the group with respect to how you handle your samples coming from all of these kind of unknown, uncharacterized environments, and what your best practices are with respect to biosafety level. On one hand, you know, like what is the best safety practice, which is sometimes aligned and sometimes not aligned with what our institutions consider to be the best safety practices. So I've been just in a battle with our EHS, for example, just to be able to handle these samples benchtop, even if they've been stored in inactivating agent, for example.
That doesn't make any sense at all. I don't understand your EHS group, because we get samples from all over the world and we put them in 95% ethanol. That reduces their BSL, BCL level to 2, and then we just handle them in a hood. So I don't understand. Do they think there's some organism that's going to pop out and get you? What did they give a justification?
Yeah, I guess. I mean, they keep asking me for like lists of organisms. You know, what are the known pathogens in your samples? And then we get into this circular argument where I say, well, actually, I don't know, because these are environmental samples. And then we just like go into this ping pong back and forth.
Is that why they don't want you to culture, or is that
not even for culture, just for nucleic acid extraction? So we're not even talking about, you know, about culturing or even, you know, even enriching in any way. You know, we're not doing any kind of viral enrichment or concentration or anything like that.
That's really bizarre. I don't know how people feel about it, because we get stuff and it's approved by APHIS, and APHIS governs a lot more than Cornell's environmental safety group. So
yeah. Yeah, it's been an uphill battle, to be honest.
And pasteurizing and alcohol they don't care about. So like going to, you know, 80 degrees centigrade isn't also acceptable. You're caught in a rock in a hard place.
That's a good question. I haven't offered that particular treatment, but because in my mind that would just denature a lot of the nucleic acids that we're interested in. But maybe if it's a short enough period of time, it doesn't matter.
No, it's better than not analyzing it at all. So I mean, obviously you have anaerobes in here, right? What percentage are anaerobe versus aerobes? Because it looked like you had a bunch of anaerobes, right?
Yeah.
Because sodium azide isn't going to do the trick for the electron acceptors for anything but things that are under aerobic respiration. So how do you deal with anaerobes? But boy, 95% alcohol, you would think, and they aren't accepting DNA RNA shield, right? Even though that's guanidinium plus TC, or what is it, TTAB.
Yeah, and has all the FDA approvals even for SARS-CoV-2 inactivation.
Good luck with that.
Yeah, I know. So yeah, various members of the group have been helping me in this process. So hopefully soon we'll be able to scale this up a little bit.
So as Stefan just echoed, heating to 9095 and doing 1s libraries in case it denatures into single strand is still an option. The question I would have to that, Stefan, is if you heat something to 95, is there some chemistry that actually could cause hydrolysis of the DNA? I think is what you were thinking, right, Elizabeth?
Mm-hmm.
I mean, do you worry too much about that if you're doing shotgun sequencing? You might not, even if you fragment your DNA somewhat. I mean, it's able to handle 95 degrees anyhow. I do think that denaturation is the biggest issue. Then you wouldn't be able to do a usual double-stranded DNA library prep from it. But if you use one of the single-stranded library preps, then you can still get shotgun from it. Yep. And then hopefully that would, I'm with Scott on this. I think this is insanity. I mean, if it's in DNA RNA shield, like what are we worried about?
Yeah.
Sorry, Elizabeth. That's your new life now. But I did have a question about.
It would be easier to do this in my kitchen.
Maybe. I don't want to come over for dinner though.
Yeah.
Gowanus burgers. Did you do long reads? And I wasn't sure how much, because I'm going to lead into the next subject matter. Did you do long reads? I know that you guys do long reads, but how much long reads did you do on this data? And did you pump that data into your biosynthetic gene cluster pathway analysis?
We did actually some nanopore reads on just a couple of the surface samples that we had as kind of a trial. But this was a number of years ago, actually. So I imagine the quality that we would get now is different than what we were getting at that point, when the error rate was still pretty high. So we kind of did some trickery where, we corrected the nanopore reads with the Illumina reads to try to like bring down the error rates. But what was cool is that we just did like a really small subset, but what was cool is that we're able to identify some regions that spanned both genes of interest and also like species specific markers that allowed us to pair together those two disparate sources of information. So yeah, long reads is definitely very useful for these various research questions that we've got here. So excited to hear what Concetta has to share with us.
So without further ado, I've asked Concetta, maybe I'm saying that wrong, I'm sorry if I am.
No, it's fine.
To come and give an update on Oxford Nanopore. A lot of us use that technology who on the call right now. And I will tell you the software updates happen so fast sometimes, and the methods are updating so fast that it seems like every 3 days I turn my head and there's a whole new method that I should be using that I'm not using yet. So I said, I would love for you to give us an update. And I think I kind of give you a little bit of background of what people might be interested in, but please feel free to discuss whatever you'd like to update us on long read.
Thank you for the opportunity. And I try to follow your guideline, try to pull together the latest and the greatest from Oxford Nanopore. So just let's cut short on introduction. My name is Concetta, and I'm based in New York office. So I'm the technical manager for the North America. So as Scott was basically mentioning, as a company, because you're already a user, you know that we are relatively young. So we came in the market 6 years ago with just one product, MinION, one chemistry, and that was it. So in 6 years, we have been having this kind of exponential growth, and we have diversified our portfolio. But we are still growing, and we still believe that there's a lot of room for improvement for us. Here, what I want to capture is basically in a snapshot all the continuous innovation and improvement that we have been basically coming along in the last year or so. So whenever Oxford Nanopore basically announce any improvement, we usually try to tackle that on different angles, from the platform per se, and then we go on the chemistry with the pore in the flow cell or the chemistry, and then with the algorithm and with analysis. So basically, all together, these aspects they make Oxford Nanopore innovative. So, and today I would like to basically to go through each of kind of these 4 segments of improvement that we have been having in the last year or so. So MinION, I think probably a lot of you have started using our platform, using the MinION is our easy plug-in and sequence device. We came with this kind of need of a sort of it's not even second generation, because we had a minute kind of intermediate device sequencing with the Mark 1C. Mark 1C is our, we call, 3rd generation of portable device, because it's a compact sequencer where you don't need anything else than just go out in the field and sequencing, because as the monitor is already encapsulated with the MinION, is also on Wi-Fi, so basically you don't need to have internet access. The advantage of the Mark 1C is basically the GPU capability, so it's more powerful than a MinION basically plug it in your own computer. And here are basically the computer spec here, 6-core ARM processor and TX2 GPU. So here few snapshot from people that they're taking this device in the field. Alexander Wittenberg from VIB is known in the field, in agri-food field, and then Render Danielard. So they use our device quite successfully. Now, the other innovation that we launched was basically the Flongol. Flongol is flow cell in a smaller kind of size in terms of pore and throughput generation, where we lower the cost of flow cell compared to the regular flow cell. So in this case, you can optimize the cost for run, especially if you're using Flongol for quick pathogen identification. So you have the dongle where all the circuitry and the ASICs is basically embedded, and the consumable per se is basically this top part here, just I will call a piece of plastic, basically, where 126 ASIC channel are seated. So people have been, Flongol has been in kind of that early kind of access development phase, and at the beginning, we have been ourselves, using a little bit of extra time to optimize basically Flongol usage. Right now, we probably, if you have been following some of the updates, now we have a specific expansion package for the Flongol for the priming and loading, and now the performance of Flongol flow cell are pretty much stable where we want to be this product. And people, they use Flongol very successfully across different type of application. They are able to, have been able to generate up to 1.5 gigs of data in 12 hours, has been used successfully also for some target sequencing with Cas9 with 100x enrichment. So it's a pretty valuable, I will say, piece of equipment that we have put on the market. And you can use use Flongol either on MinION, Mark1C, or GridION. Now, talking about sample prep, Scott has asked me also to cover something about the Voltrax. There are Voltrax is our automated in a small scale library prep. So where a library prep that goes for instance delegation which average around an hour or so. With Voltrax you can basically complete that in the near future in a much shorter time. The advantage of the Voltrax is that you reduce a lot that kind of variability of the manual library prep operation. Now, on the right-hand side here, I just posted some of the latest update because we are upgrading the Voltrax user with the V2B device. There has been a new Voltrax software update in March for fixing a few bugs for the cartridge configuration one. But most important is basically to prepare for the launch of the new chemistry. One of the new chemistry has been launched is the multiplexing. And you have here, you can see in the store, I just got a screenshot on the bottom. You have the you are able to multiplex up to 10 samples. You can see here VB01 to 10. Those are the 10 vial with the unique barcode, and the remaining of the tubes are identical to the single plex. This chemistry is slightly different from the previous one. There is change in volume requirement and new formulation with a new oil, and this in preparation for the big launch that will be for Voltrax, which will be a PCR-based type of chemistry and feature that is will be enabled into the device. Now, moving from, let's say, the chemistry now to the flow cell. So we have two main type of pore that are embedded in our flow cell consumable. So the R9 type, and we call it commercially flow 106 when you receive your flow cell, and then the flow 110, which are basically the R10. The difference across the two are, you can see from the scheme, this transversal basically picture. Oh, sorry, there was some echo. Is basically that the R10 have this kind of double head, and they've been kind of engineered with the scope of try to resolve one of the probably main challenging for the single read accuracy that was for Oxford Nanopore at the very beginning, basically the homopolymer region. So with the double head, you are able to cover a much longer stretch of homopolymer and therefore calling with higher accuracy than a single strand. You can see here some quote from people in the field, the customer, they've been adopting the R10. People, they are in the field of HLI typing, so they were able with the R10 to get the high resolution 2 and 3 field typing, and people that are using basically this for metazoa sample, reaching a very high consensus then accuracy at the very end. And here showing basically how the accuracy moves across the stretch of homopolymer. The purple one is basically the R10 type flow cell. You can see you can call with quite good confidence a stretch homopolymer, up to 6 before start to kind of experience a little bit of decrease, a few points of decrease in single read accuracy when you go 7 or 8 in terms of homopolymer length. Continue on the same kind of talking about accuracy improvement. Not sure if you have heard about one of our new product that is in early access. It's called the Q20 chemistry. So the Q20 chemistry will be not only a chemistry per se, but a combination of the chemistry and also algorithm-based calling. So with the Q20, we are trying to reach that 99.9% of accuracy on single reads. So we have opened this early access registration. There is a small group already that has been testing, and here are some of their quote from Matt Luz or from Rasmus in Vienna. So you can see how the shift from the previous type of chemistry in human and the Zymomoc community, if you compare top and bottom. So you have basically this modal single accuracy now that is basically shifting past the 99 percentage. So we are 99.3 for as a model here and in human, and close also with the Zymo. And here a snapshot from GBF graph. Now, all this is, as I mentioned earlier, comes basically together with work that comes from multiple, departments. So we look on the chemistry side, but we also look on the algorithm. So there've been new improvement, and I think now just to go back to Elizabeth. So 2 years ago for Oxford Nanopore is basically kind of a little bit ancient, because we have been kind of improving so much all the component of our platform. So now we have a new algorithm and a new base caller bonito, which increase that single read accuracy that I've been talking now. All these are embedded in our software that we deployed. There is Guppy standalone, and then we embedded this in the Minnow software release that is packaged for each device, for MinION, for GridION, and for PromethION. And I want to move just right away in that continuous improvement of the raw read accuracy. And you can see on the right hand side here, this basically graph where it's showing basically the history of our improvement across the last 2 years and across a number of update of the software. We had for the longest time Albacore as our base caller, and has been retired summer of 3 years ago. Since then, we employed Guppy, and Guppy has been going through a serious number of improvement. So changing time to time the type of base caller from the transducer model to the flip-flop, and now we have our Bonito model that is basically embedded in that CNN, the convolutional neural network. So basically, there is not only there is a sort of a prediction in this algorithm of not only the base at called, but also the neighbor that is basically around that specific base. For those of you that are into software and development, so there are a number of research-based base caller that are available on GitHub, each of them with their own kind of flavor of improvement. So we have Runny, that is research-based caller that try to target specifically the homopolymer calling. Then there is Bonito for higher raw read accuracy, and then there is Rario, which is sort of a repository of different base caller models. So you can, through GitHub, basically be part of this development or developers group and start to train your own base caller based on your own research model. And here some more specification of what each of them are. So as I mentioned before, RANI is basically for homopolymer. All of them, they are looking after the raw signal, and based on the algorithm that they are built around, they do their own kind of prediction of the raw signal input. One of the other information that I want to rely on you today is basically what is embedded in the base caller in the device that you have on available. So on Gridiron, you have Minot, and same Minot packaging is basically in the MinION. So one of the latest update we have been kind of, which is I think was probably a month or so ago, we have been improving the accuracy of the different models that are embedded. So there are actually three. There is the fast base calling, which has a raw model accuracy of 95.8. The high accuracy, which gives 2 points in accuracy higher. And now there is this third one, a new one, that is basically called super accuracy. So which reach basically at model distribution of accuracy around 98.3%. Now, what also I would like to point out, you can see there are difference also on the accuracy if you consider not only the model but also the type of flow cell. For the R10 type, there are few percentage lower for single read accuracy compared to the R9. And the other thing to take in account is that the SUP one is one that require more is more computational involved. So the base calling, even on Gridiron, which has a GPU, you will experience some sort of lag in real-time base calling, and this kind of decrease as you move down from the SUP to the fast. So fast is the model that allows device as a PromethION and Gridiron to be up to speed with the live base calling. As you move in that high accuracy, you move in the requirement of having more computational power involved, that real time start to decrease a little bit. Here, kind of showing where we are with these early access data for the Q20, showing the model distribution of the quality score. This new chemistry will be quite different from the current LSK 109 or LSK 110. There is a new motor protein which will work at a different speed of translocation. Will still be compatible with the current flow cell type. And here some snapshot tweet from who has been helping us in testing in the early access the Q20, Meet and Jay from University of California, Santa Cruz, and always Alexander Wittenberg is one of our kind of strong customer in the plant field. Here, changing a little bit, shifting a little bit of kind of interest and moving towards the SV calling. So all the improvement that we have been achieving have been having as a kind of main goal the tentative to have an improvement in two of the major analysis pipeline, SNP and structural variation calling. And you can see here what we have reported as a benchmark data. We have been kind of having an improvement in precision and recalling. And as you go up with the coverage, you basically improve those numbers, those stats, and there is basically an improvement of the false kind of positive call rate as shown here. And here the graph reporting basically the recall number trend versus the precision one. another graph to basically kind of summarize what mentioned right now in terms of the benefit of our tools, not only because we are improving in the software algorithm, but also especially with structural variation, long reads are definitely beneficial for be able to span long, repetitive, and big structural variation point in the genome. As mentioned earlier, so the improvement are all on basically on the double front, also on the SNP calling, and we have been kind of combining our Medaka tool with the Google Deep variant. There are public data set available on GitHub, so you can download and test those data set on our new improved algorithm. I see that there are a couple of questions. I don't know if you want me to start to address now, or we can do everything later. I'm almost kind of halfway through. I'm going to move.
Happy to address. You can address them as you see fit. No problem with that.
Okay, because we are just now moving to the consensus accuracy, moving from the single read accuracy to the consensus, and now of course you can imagine, so by improving the single read accuracy, so the single read error rate, so you improve basically overall the accuracy of your consensus. In here you can see the what we have been reporting in terms of Q45 and Q50 for the two different type of flow cell type, and this has been benchmarked across different number of bacterial sample coming from the Zymo community. So the pipeline is pretty simple, kind of straightforward. So you use the most the latest Guppy base caller, then you assemble it with Fly, and then you polish with Medaka. And here showing the difference by using the three different model that I mentioned you earlier. So fast versus high accuracy versus super accuracy. You can see you have the improvement of that consensus quality going basically towards the 50 when you use super accuracy. Another thing I want to mention, I forgot to mention before, because now I see in front of me the three different base caller. In the latest software update through Mino for Minion for Gridiron, there's been a change in the default quality score, where until now the quality score for passing read was a 7. This basically threshold has been kind of, the bar has been increased. So for fast, the quality score is basically more than 8, 9 for high accuracy, and basically 10 for super accuracy. So we have been kind of getting much more confident in the way the raw signal get basically base called. And now we go to quickly on the methylation, just 2 slides to, one of the features, as you probably may know with Oxford Nanopore, is that because you're sequencing the naive molecular nucleic acid, so basically you have the natural signature that are embedded, and you don't need to perform, very long laborious type of wet lab library prep with the bisulfite conversion in order basically to address a question related to methylation. So, and we have improved our base call system as well, and we have incorporated also the methylation detection while you basically base calling while sequencing, and this is in Megalodon. right now, what we have available, as you may know, are kind of the most the classical and well-known characterized, methylation side is 5-methyl-C and 5-hydroxymethyl-C, and then 6-methyl-A. Yeah, these are the information that I just convey. Now, going on the chemistry, we have, I mentioned, LSK 109 as a library prep. That is the ligation based. In November last year, we announced a new chemistry, LSK 110, which is supposed basically to replace LSK 109, which will be superseded. But considering the COVID pandemic and a number of additional protocols that we have been rolling out, they are based on that chemistry. LSK 109 is basically running in parallel side by side with LSK 110. Now, what are the advantages of LSK 110 and why basically will in the near future oversee the LSK 109? There are sort of improvement in the chemistry formulation. I have basically highlighted with this arrow pointed here. There is a new adapter mix in fuel in the adapter tube. So because we'll support the consumption of that fuel, that ATP, when you have the motor protein basically ligated to your nucleic acid, to the basically the end. That consumption basically starts to kind of kick in, regardless if the platform is sequencing or not. So we have been implementing that aspect, and at the same time, this chemistry cover a little bit much wider range of fragment size. So it goes basically from very short library prep to the ultra long reads. For this specific reason, because now in this improvement of the sequencing buffer and the fuel, you don't have that phenomenon when you sequence short reads that you see the speed of translocation basically dropping, because you have been depleting the sequencing from the fuel, from that ATP. And therefore, when you decrease the speed of translocation, that will affect also the quality of your reads. So here is just kind of short list of what I mentioned, the what is new and the improvement of the LSK 110. There's the same number of reactions, same price. The protocol has a slight modification, but overall, in terms of hands-on, is similar to LSK 109. At this particular time, probably one of the downside of pitfall I will say is that the LSK 110, we have not finalized the optimization of the multiplexing aspect with the native barcoding for LSK 110. So the only multiplexing option available is the one by PCR. So including the barcodes up to 96 by PCR. And as in the note I wrote here down is basically we are continuing to support LSK 109 because we have a very important segment of our market that is covered for the COVID, that is covered by the LSK 109 and the native sequencing multiplexing. A new kit that we launched, I think, early this year, towards the end of winter, is the ultra long kit. So this has been a kind of successful collaboration with Circulomics. Circulomics, I'm not sure if you are familiar, you have been using already their chemistry. They're kind of specialized in extraction, high molecular weight genomic DNA. So we've been kind of partnering with them to combine the extraction, which is one of the key factors for be successful for Oxford Nanopore platform and with ultra long reads. Here you can see some of customer experience on their usage with ultra long kit and with circulomics. So basically N50 are very high and with very high long reads. Glennis is part of the Ecker lab in University of Washington. They've been part of that big consortium with Karen Megan for the T2T2 and part of that consortium that just basically closed the gap of the 8% of missing human genome sequencing. So they've been a big contributor with Oxford Nanopore for this milestone result. So here in terms of library prep, the library prep with the ultra long kit is a little bit longer because you want to achieve these ultra mega long reads. So it takes overall the library prep per se more than 3 hours, and then there is an overnight kind of elution step before you are able to finalize the last part of the library prep with the ligation of the motor protein and start sequencing. Then, as you know, the sequencing run, depending on what is your end goal, can last basically anything in few hours or can be spent in days, so up to 72 hours. And here some of the result using the ultra long sequencing kit, because one of the advantage of this chemistry is also the fact that all the attempt by using different extraction methods, they always result in fragmenting the DNA. And then, if that is fragmented, of course, your output is going to reflect that aspect. On the other hand, working with ultra long reads is also challenging because they may try to tend outside the flow cell, the pore, some sort of secondary structure. So a combination of the new improved washing kit is used when you run this ultra long sequencing protocol. So basically you monitor your run and you start basically washing and flashing and adding more library prep. Basically from each library prep you can do in average 3 loads in this kind of way. And you can see here the representation when you see starting basically plateau, the type of throughput, that is basically the good time when you can wash and basically reload your flow cell. And that will be beneficial for overall the throughput generation out of the 72 hours run. Here, just an additional graph that is showing some of our internal data on human cell line with a throughput of 24 gigs in kind of ion flow cell and 50 close to almost 100. If we go now on the multiplexing, I know from Scott that he just recently came aware of our increased multiplexing option. So the rapid barcoding kit RBK 110.96. So it's basically a 96 multiplexing option using the rapid chemistry. So this is the chemistry that is at base of our midnight protocol for COVID sequencing, and it has been available, made available the last month or so. We have expanded our native barcoding kit. Now we have 96 native barcoding available. As I mentioned before, those are right now only compatible with LSK 109. We haven't finalized our optimization protocol for 110, which you can always still rely on multiplexing option by using the PCR barcoding. I think this is my last one, so I can stop sharing and I can now try to address some of the question.
I was just a little interesting. This is a really good update. Really appreciate it. But so the last slide you just showed, it looked like the first line said LSK 110 was good with NBD 196, but then the next slide said it wasn't compatible with NBD 196. And I'm just wondering, is NBD native barcoding compatible with both LSK 110 and 109?
No, if there was that, probably my bad in kind of rushing at putting together, the native barcoding NBD is not compatible yet with the LSK 110. So
Okay.
you still, yes, you still go with the PCR for the LSK 110 chemistry.
LSK 109 plus NBD 196 is a go then, right?
Yes, yes.
Okay. Questions, comments, technical discussions about Nanopore. Open floor to the group.
Yeah, I have one question. I was hoping to get some updates about adaptive sampling or read until. I know at the MK2 it's already implemented, but I heard that a lot of people, not a lot, a couple of people have problems to install them on their clusters.
Yes, because the adaptive sampling is has been kind of rolled out because it's computational intensive as a process. So as first being rolled out on the GridION. So on GridION, you can basically do adaptive sequencing as you go, as you sequence. The same component has been embedded in Mino release, which goes on the Mark 1B or Mark 1Z just more recently. So there probably is not still fine-tuned for the installation. Is less practical for that compared to the GridION, but is working. So, and actually in the latest release there are some adaptive sampling improvement for the CVS, the output of the CVS file. So everything should be good to go now. And again, this is good across the three platforms at this point. So basically adaptive sampling on Mark 1C, Mino in MinION, and then in on GridION.
So I got a quick question. So can we run 16S full-length libraries on the Flongle? I didn't check the GridION, but I thought I'd ask right now.
I don't see. To be honest, I don't recall top of my head if we ever made a protocol optimization for that. But considering that the 16S as basically that a rapid attachment, so I won't see a big problem on running on Flongle, because on Flongle people, they run the rapid chemistry. So I will say no. Since we are here, let's see if I check on quickly. I have the other monitor open if we have the protocol for Flongle. So on 16S. It's more like for Flongle, see if what level, what type of depth of sequencing you want, because as you know, with Flongle, yes, we have 16S for Flongle. So
okay, great, because even if you barcode 12 samples and put it on there and get even 800 megs, that's a ton of coverage. So I just turned on the GridION this morning to look and see if it was on there. But cool, thank you.
Yes.
Any other questions or comments?
One thing that, since we are touching Flongle, one thing I wanna kind of bring on the table is that when you use a different flow cell type, Flongle or the R9 or R10, just make sure to follow. There are some guideline in terms of the amount of input material and the library prep that you load there, because that will kind of affect the performance. So you need, of course, to load less in Flongol and more in the R10 flow cell versus, the R9. There are different requirements.
Well, thanks, Concetta. I appreciate that. That's a really good update. As I get, just so everybody knows, I asked Concetta and Andrew last night at 6 p.m. if they would do this update. So this was literally last week. So thank you very much for stepping up to the plate on that.
You're welcome. So as I said, I will now let you continue with your meeting. We can set up, kind of really a quarter kind of update, and we can, do stuff like today with also specific topic requirement. We can probably, coming with a list ahead of time if you have specific, interest, and we can try to cover those.
Cool, thank you. All right, so we're going to move on to our last brief update. I'm going to hand this over to Horia, JJ, and Krista, just to kind of briefly go over what we did before on the samples that came over from Romania. We kind of used a different storage approach to get samples over here. And I think, Horia, did you want me to share the screen with you, or? Yeah.
Actually, I would like to ask you the permission to share 2 slides, just to brief introduction to our the aim of research. And then I will ask you to allow Andrea to show her sample preparation slide, okay?
Yeah, let me see, hold on for just 1 second. So there's still plenty of people here. I gotta kind of navigate and find you. There you are. Okay, Horia, you are co-host now. You should have control.
Okay. I will try to start sharing. Do you see my slides?
Yep.
Yep.
Okay, just briefly, we want to sample actually 2 lakes. These lakes are hyposaline. And one is a natural hyposaline lake, and the other one is kind of mine pit lake. You see, that's a kind of upside down bell shape with a very high depth. So what are we looking here? You see, both lakes are more or less water stratified. So they have 3 water masses. One which is the uppermost water masses, which is less saline. Then we have a transition zone, which is called chemocline, and a deeper zone, which is called monomolymnion. So we took depth-wise sampling from this part, from upper and lower part of the chemocline. So we want to cover the vertical, the water column, and two points here in the depth, in the higher depth of the lake, and an extra point in the actually at the bottom of the lake from the sediment. So we end up with 12 samples. That is 6 samples from one lake and 6 samples from the other lake. And in the end, we wanted to compare the microbial diversity. And by long read sequencing, I told Scott that we are very interested in recovering DNA from eukaryotes living here in hyposaline water mass. And this water mass is also a kind of sulfidic and anoxic. So it would be very, very interesting to find eukaryotes living in such condition. Okay, so this is my introduction, and I will try to get out of here. Okay, and I ask Scott to allow Andrea.
Sure, sure. 1 second. Let me just find him here. And then...
Hi everyone. I just have 2 slides.
Ah, got it. Good. You should be able to share the screen, or or can you
One
share
moment.
yet?
Let me see. Let me see.
Do you have control?
Share screen.
So until Andrea is ready, I want to tell you that all this sample are analyzed by short read sequencing, shotgun sequencing, metatranscriptomics, amplicon sequencing, and even metaviromics for the deepest sample. So we want to make it very big and very, I'd say, very detailed analysis of the biodiversity in the lake.
So can you see my slide?
Right.
I think so. Yeah. So we use the 0.2 micrometers membranes for water sampling and for water filtration. And for the sediment, we had a Poner grab to collect the sediment. I think from Ursul Lake, it was more than 11 meters, the depth for the sediment sample, and I'm not sure, but in for a Fund Lake, bottomless, if you want a translation, it was around 27 meters. So, and for the extraction, we used the Power Soil kit, DNA Easy. You recommended this kit, and we stopped at the before the elution step. So that was all we did with these samples. We had only one membrane, so one column. And it's in these lakes, it is very difficult to get a large amount of DNA because we cannot filter a high volume of water because of the high salt concentration and also the biomass, microbial biomass, algal biomass in the water sample. So we have volumes in the range of 50 milliliters to 600 milliliters on one membrane, depending on each water strata sampled. So that's why I think we have variation in DNA concentration. Maybe JJ can show us the data related to DNA concentration.
Yep. So if we're wanting to transition, that will be Krista, who did the work of eluting sample. That's right. Yeah, Krista did the incredible job of eluting the samples. So something to note, so Scott and Horia knew this, and I don't know if you knew this, Andrea, but the samples stayed on the columns for longer than maybe they should have, or we anticipated to. But what was interesting were the results that we had, which Krista can show. And I'll admit I had not received samples that were stored on a column before, so this was new to me. And so yeah, Krista, if you want to show them what we found.
Everyone see my screen?
Yep.
Sorry. Yeah. So as Horia, JJ, and Andrea said, we received the 12 samples at our facility here in New York, and they did stay on columns for quite a bit, but we were able to start processing them. As suggested by Scott, we did leave them at room temperature for a little bit with some nucleus-free water, and then we centrifuged in order to remove from the columns. And then afterwards, we did a DNA concentration for high sensitivity to see what we could pick up. And we were able to pick up some DNA, and then we did run it on the tape station to see what type of quality we have in the samples. So moving forward, it looks like we'll be able to process a set of these samples. And that's what I have.
Let me ask this: when you say out of range, what does that mean? Is it too low or too high?
Too high.
Oh, okay, so you didn't dilute them and rerun them?
So we, we're going to do another test on these samples. We're waiting on a kit to arrive, I think we're going to do a broad range qubit just to see if we get any other results, but this is what we have now.
Oh, okay, you just don't take it and do a 1 to 10 and just run your dilution. But I will just say, that's a ton of DNA.
Yeah, it's plenty.
So yeah, I think we're good.
Yeah, this is phenomenal, and the molecular weight is really amazing. And I just want people to know, so this isn't my idea. This is stuff that people have been doing all the time, where they take their DNA, they put it through their capture columns, and instead of eluting it, they just spin it really well to get rid of the alcohol rinse, and they take those columns and they ship them out. And usually you try to elute the DNA off in 5 millimolar or 10 millimolar tris, pH 8 at 50 degrees centigrade within a week. But the beauty of this is you guys let them sit for a really long time, and then you even elute it in water, right? So how long were they stored in exactly? Do you know? Is it more than 2 weeks?
I would say more than 2 weeks, yeah.
Wow. Wow, this is amazing.
Is that good news, Scott, in your view?
No, this is great. I mean, we never tested it. We just take it, put it through, and try to get it on the other side of the country, and then we elute off in warm tris, and we usually get pretty good DNA. But I've never let it hang out for like several weeks, and this data is super high molecular weight.
Yeah, so educate me a bit, because I've never received the samples on a column. What's the benefit of having them on the column or leaving them on the column after the wash?
Yeah, so think about it. What we got is a isothianate salt bridge that's binding to the DNA electrostatically. So it's not covalently, it's just electrostatically through a thiosulfate bridging to the DNA. So one is we'll keep the DNA fairly stable and elongated and attached to the silicon. And the last thing to touch is alcohol. So the chance of getting DNases in there are like virtually nothing. So you're stabilizing the DNA structure, you're holding it there through salt bridging, and then the next thing you do, it shows up on the other side of the planet, you're eluting it in tris. So, but by accident, you guys had let them sit for several weeks and proved to us that it doesn't seem to matter. You got tons of DNA, it's fairly long DNA, and you haven't sequenced it yet, or you have?
They are on the docket to be sequenced. So
They're going soon. The other thing that's happening when they're on the column like that, they're dry, and basically they have no water, so you can't have a nucleophilic attack from water, which is what GenVault, there's a company that just talked about storing DNA or even RNA on just dry cards instead of freezers, because if it's dry enough, it's actually pretty stable. So I think maybe that helped too.
Yeah, I think like exactly, Chris, this is Danny. I think like the combination of the high salt ethanol wash and dry out membrane is exactly like these stances, what you can keep up to 12 weeks in postcards sending around. And also one question is, did you remove the RNA with RNAs? Does the kit has RNAs in it?
Probably not yet.
Yeah, that's Andrea or Scott.
Yeah, no, that would be Andrea or Horia can answer that.
Just let me look at the protocol.
Yeah. But I will tell you the beauty of not putting it on an FTA card like GenVault does is that your next step is just adding tris and you're done.
That's great. One quick thing I might announce, I have to hop to another meeting at 5, but we are thinking about having a meta-sub meeting in October, it looks like, in Miami, and we could invite everyone from XMP if they want to come potentially. We'll know more certainly by next time we have a call, but we're trying to finalize the details for that to get together in person. Ideally, everyone should be vaccinated, of course, and hopefully people are starting to feel like it's okay to travel, but it depends on personal choice. We'll also broadcast the meeting, but as a kind of a sister group to the XMP is the MetaSub group, is we're gonna have hopefully a meeting in Miami in October, like around Columbus Day weekend, or that week right after, it sounds like. So and everyone's welcome. That's a quick note.
So as I remember, there is no RNA removal in the DNA kit. We did RNA extraction at the same time, so we have DNAs there, but in this kit there is no RNA removal. solution, at least,
So
if I remember correctly.
You have also, maybe you have to check also if the RNAs if you find any RNA, that would be absolutely like gold if you have like full nucleotide stable sample on a filter, not only DNA, but also maybe a bit RNA. Maybe because of the high salt concentration, it's fine. Who knows?
We haven't tried that yet. We have done it internally through ABRF where we've dried, and if Streeter's on the call, he can kind of contest to this, but we have taken RNA, stored it on an RNA column and shipped them around the country, and it worked really good as well, but we don't have any, I don't think we've written a paper on it anywhere.
Yeah, also wondering if the kit what you're using, remember, Scott, we discussed once that there is in the mobile, we have like C3 and C4, they're removing organic and inorganic compounds. In those samples, I'm pretty sure not. So that means whatever is in the seawater, maybe that there is some metabolite compound salt, special salt, which also stabilize the sample. So you definitely should maybe take the absolutely 100% clean DNA free of any salt components, salt metabolites, and then do this experiment so it's still stable 12 weeks on these filters.
Yeah, it might not be every single DNA or RNA kit that works. So we know this one works because you guys just verified it, but okay, we've gone quite a bit over, and I'm really sorry to keep everybody extra long time. But I will be recruiting other speakers for next month. And if anybody has any closing comments or statements, otherwise we will close the meeting. All right, well, thanks everybody for attending, and we will see you in a month.
Okay, thank you for help with the long read.
You're welcome.
Very excited to see the results for you.
Thank you, Chris.